Now let us examine the structure of a sperm. It is a microscopic structure which is composed of a head, neck, and a middle piece and a tail. Plasma membrane envelops the whole body of sperm. The sperm head contains an elongated haploid nucleus. So head में होता है nucleus which is haploid and the nucleus is elongated. The anterior portion of which is covered by a cap-like structure which is known as acrosome. An acrosome is filled with enzymes that helps in the fertilization of the ovum. The middle piece possesses numerous mitochondria. So M for middle piece, M for mitochondria, which produce energy for the movement of tail, thus facilitate sperm motility, which is essential for fertilization. The human male ejaculates approximately 200 to 300 millions of sperms during a coitus, which for normal fertility should be at least 60% of the sperms must have normal shape and size, and of that 60%, 40% should be at least motile, vigorous motility show करना चाहिए. Now the sperms released from the seminiferous tubules are transported by the accessory ducts. Secretions of epididymis, vas deferens, seminal vesicles, and prostate are essential for the maturation and motility of sperm. So this is very important. Question can come, which are the ducts? Secretions of what can help in the maturity of sperm? So epididymis, vas deferens, seminal vesicle, and prostate. Bulbourethral is just for lubrication. So the seminal plasma along with the sperms constitute the semen. The functions of the male accessory ducts and glands are maintained by the testicular hormone androgens. The process of formation of a mature female gamete is known as oogenesis, which is markedly different from spermatogenesis. Oogenesis is initiated during the embryonic development stage, when a couple of millions of gamete mother cell, that is oogonia, are formed within each fetal ovary. No more oogonia are formed and added after birth. So these cells start division and enter into prophase 1 of meiotic division and get temporarily arrested at that stage, which is called the primary oocyte. Now each primary oocyte then gets surrounded by a layer of granulosa cells and is called the primary follicle. A large number of these follicles degenerate during the phase from birth to puberty. Therefore, at puberty, only 60,000 to 80,000 primary follicles are left in each ovary. The primary follicles get surrounded by more layers of granular cells, and a new theca are called secondary follicles. So the secondary follicles soon transform into a tertiary follicle, which is characterized by a fluid-filled cavity called antrum. The theca layer is organized into an inner theca interna and an outer theca externa. It is important to draw your attention that it is at this stage that the primary oocyte within the tertiary follicle grows in size and completes its first meiotic division, and it is an unequal division resulting in the formation of a large haploid secondary oocyte and first tiny polar body. The secondary oocyte retains the bulk of the nutrient-rich cytoplasm of the primary oocyte. Can you think of any advantage for this? Thus, the first polar body born out of the first meiotic division divide further or degenerate. So we basically do not know till now what is the function of the polar body. At present, we are not very certain about this. The tertiary follicle further changes into the mature follicle or the Graafian follicle. The secondary oocyte forms a new membrane called zona pellucida surrounding it. Graafian follicle now ruptures to release the secondary oocyte ovum from the ovary by the process called ovulation. Can you identify the major differences between spermatogenesis and oogenesis? A diagrammatic representation यहाँ पर दिया हुआ है just to find out the differences.